Perfect. I actually have a funny story. A guy I knew didn't shut down one of these web instances,
Oh no.
and it went for like a week, and it was $60,000.
No.
So that's no joke. What you said, if you leave them open, you pay for it. Thankfully, they let them off, but it could have been very bad.
Huge American money.
Okay. My computer updated last night, and then it completely blew up. So do you know where the remote is? Okay, here we go.
Oh, no.
This is a Mac feature. Is that working? Okay, cool. Hello, everybody. Thank you all for inviting me down here. Up until just right at the beginning of the year, I moved my group up to Lannel. But I have a joint appointment still on North Campus. So my PhD students are still there. So I still live very much in the UNM world. But yeah, so my group literally couldn't do its job without Varsity Resources, and that we do really large scale pathogen genomics projects. So that's lots of data and lots of computation. And more, we're trying to do these things faster and faster to do real-time insights. And so I'll just give you a whistle-stop tour of some of the projects that we've been working on. We did a lot of SARS-CoV-2 work. We do a lot of diarrheal disease. We joke like we're like the waist down, like infectious disease group. And a lot of work that we're doing looking at sort of hospital-acquired infections. So my group uses genomic data and lots of it to really answer epi questions with that data. And so those tend to be things like what's causing an outbreak, something that was very important in about like December of 2019. When and where did an outbreak begin? How many introductions have there been? So like how many times have we seen a pathogen come into an area? And that can be like at any geographic scale. It can be continents to wards in a hospital or beds in a room. How are outbreaks or cases linked? So do we see transmission? What is the resistance profile? So like, are the drugs that we're giving to treat actually effective for this pathogen? And one that we're probably all now very sick of hearing about. Actually, we don't even hear about it anymore. SARS-CoV-2 is also on the don't talk list. So how's the pathogen evolving? Like new variants, right? So, Delta, Omicron, all these things. And yeah, so we're using fundamentally genomics data. And so we're of course looking at DNA and genomes from viruses, bacteria, and fungi. I joke we don't do anything diploid that's too complicated for me. I need haploid organisms. so no double strand stuff. So so we're looking at sort of these molecular fingerprints, and then typically we're using that data, and almost always we end up with a phylogenetic tree, sort of relating how these samples are related to each other, and then we layer metadata on top of that. So I joke that we make a tree and we color it in, and that's kind of what we do for our jobs. And so I got stuck into doing this large scale genomic stuff, primarily with working with really large cholera data sets, so Vibrio cholerae, a diarrheal illness. And so my first foray into really using like high performance computing was analyzing thousands and thousands of cholera genomes from across the globe. And so my first actually foray here in using that was looking at an outbreak of a nearly pan-resistant strain in Zimbabwe. So it's only susceptible to a single antibiotic class, which is very scary. And then this continued into other projects looking at household spread in the DRC, so Democratic Republic of Congo. And of course, we can do wonderful things like end cholera by 2030. I would love to be out of a job in cholera by 2030. I don't think we're going to get there. Moving a bit more closer to Albuquerque. So I have a fantastic PhD student that I share with Don Natvig, who's in biology. And Sarah is also an epidemiologist at DOH. So this works out wonderfully because she has all of the data that we need to answer a lot of questions. And so there was an outbreak of Shigella flexneri. So Shigella is bacillus dysentery. So kind of like a cousin to cholera. So a diarrheal illness. And there was an outbreak that started sort of early 2021 and is ongoing. Actually, this is just where Sarah made me stop collecting data because she has to graduate at some point. But it's an ongoing outbreak that's primarily been circulating in our unhoused population. But it's kind of an odd thing in that, we have a whole bunch of cases that aren't in that population. But the biggest surprise of them all was that this strain moved from humans into the biopark and started to cause a really deadly infection in our non-human primates. And so most of the apes and primates became infected. Quite a few of them died. And it's really sad. I mean, we had, this was Herfinita, and this was Erie the Simang. And the really sort of bad thing about this, obviously, aside that they died, was that they're part of the captive breeding program. And so there were some infants involved and sort of breeding age mothers. And so this was a real sort of devastating thing for the zoo. So that initial outbreak happened in 2021. And then there was a second case in 2022 that seemed kind of like to come out of nowhere. So like the initial outbreak died down. The zoo thought they had a handle on things. And then this chimpanzee became positive in 2022 and subsequently died. And then we had another case in 2023. And so the big question was, like, how are all these things related? And so, like I said, we make a tree and color it in. And so, sort of the population that we find the strains in are labeled. And I guess, this is a super, what we call clonal outbreak. So there's not, it's like the same exact strain. So even though there's sort of like the branch lengths here sort of indicate diversity, we're only talking about, 30 to 40 to 50 mutations out of 5 million base pairs. So like very, very, very related to each other. And you can see that here if you sort of collapse this down to strains that are like within 5 mutations of each other, we basically just get kind of some blobs with some spikes out of it. And really what I want to take away from this is the major question. Well, so the major question was, how did the zoo become, like what's going on with this? And it actually looks like we had strains like multiply introduced into the zoo. So if you think that it was a single infection, all of the zoo isolates basically have a most recent common ancestor and stem from that and be completely related to each other, sort of like a traditional family tree. So they'd be clustered in this phylogeny. But what we see is we've got sort of the 2021 isolates up here. We've got the 2022 isolate here. And then our poor Simang Erie, he actually was part of the original outbreak, got shipped out to Texas, caused an outbreak there. We don't have any isolates from him from Texas, but he got moved back to the biopark, was PCR negative, stool negative for months and months and months. So, rigorous testing, all negative. And then he became positive again in 2023 and he had to be euthanized. So there was like nothing that they could do. He kept sort of killing his troop, which is, twice he did this, so poor guy. So it looks like we've had multiple introductions into the zoo, and we're working with the biopark to try to really figure this out. But there's like really no solid link of how the strain got in there. And sort of using this genomic data, we were also able to determine that the drugs, the first line drugs that were being used for treatment, both in human cases and at the zoo, this strain is resistant to. So that's why we had a lot of treatment failures, both in humans and at the zoo in 2021, because ciprofloxacin, sort of the first line treatment for Shigella, and all of our strains are resistant to ciprofloxacin. And so Sarah was able to, because she works for DOH, they issued a health alert for local clinicians to switch their antibiotic, their frontline antibiotic, from ciprofloxacin to a macrolide. And so Yes. Okay. Just real quick. We did a lot of SARS-CoV-2 sequencing, and I'm glad to be not doing that anymore. But you know, we could not have done it without Parsi, you know, especially at the very beginning. So you know, we've sequenced over 40,000, I think, at the end, SARS-CoV-2 samples in our group. You know, the majority of it was done on the nanopore, which for folks who don't know, to go from the raw signal from the actual sequencer to something usable for us, you know, it's like genomes for us to use for analysis. If you do it with a CPU to do this base calling, it can take like a week. So it's very slow, and you know, this was at a time where we sort of need answers rapidly. So we needed GPUs. So you went from like a week on the CPU, used a GPU, it's like an hour to do this. And so very much at the beginning, you know, Parsi really helped us out in getting access to some GPUs to do the base calling before we actually got a device that could do it in device. But yes, we were sort of the sequencing leads for a long time for a number of Mountain West states, plus Arkansas. I don't know how we got involved with Arkansas, but we did tons of sequencing for New Mexico, Wyoming, primarily. But one of the cool things, and this will highlight a CAR-C experience, is sort of the first detection of Omicron in the state. So we first received a positive sample. So one of the cool things that we had is that we got samples directly from the emergency department at the hospital there, which wasn't a sampling source that was being fed into anything else. So DOH doesn't sample there. You know, so it was sort of like our group was the one who was doing the ones from the ED. And so we got a positive sample at the hospital on December 7th, 2021. We got it in the lab. 2 days later, it was on the sequencer the next day. I remember this was over a weekend. The run finished. I was on CAR-C in my PJs at my house on a Sunday, and it was kind of like, oh crap, we have an Omicron. So I told DOH about it, and the next day, across all of the road signs across the state, we had Omicron here, booster up. I can't think of like another scenario where like your science ends up like being on a road sign. I hope. Yeah, and this was highlighted by NIH as being like, you know, a cool story for their 2022. Okay, some more doom and gloom. How antibiotic resistance is going to kill us all. So there's some estimates that by 2050, deaths due to resistant infections will actually outpace cancer. You know, we know this. We know that like as we keep applying you know, antibiotics to infections, we are seeing a rapid rise in resistance across the board. The WHO has a bacterial priority pathogen list. It has bugs on here like Enterobacterales. These are things like E. coli, Shigella, Klebsiella pneumoniae, et cetera. They're in this critical group. Oh, actually, sorry, Shigella is over. But part of this is carbapenem resistant and third generation cephalosporin resistance. Carbapenems are what physicians reach for as the drug of last resort. So if you come into the emergency room and you're septic and they don't know what's happening, they're going to reach for carbapenems. It's kind of like the Hail Mary. If that doesn't work, we're in some bad shape. So that's relevant because we had, so my group is kind of fun. We get notified of all sorts of like weird outbreaks in hospitals, and they now know to call us like Ghostbusters. So we get brought in for a lot of infectious disease outbreaks to try to figure out what's going on. And so in 2022, at UNM, we had 4 patients that were initially diagnosed with this. I've never even heard of this bug in my life before. Stenotrophomonas maltophilia, steno. And 2 out of the 4 patients died. So they came in with severe COVID, they were intubated, and they ended up dying of severe bacterial pneumonia because of this bug. Now, the scary thing about this is that steno is intrinsically resistant to many of these like Hail Mary drugs. So carbapenem's intrinsically resistant, and same with those third generation cephalosporins. And so Acinetobacter is sort of like one of these really bad guy sort of super bugs. And all of these are all the drug classes, and the R's stand for resistance. Look at steno. It's like, thankfully, it's not very virulent, but it is hell resistant. I won't go through sort of so this is the picture of the ICU and how things were related to each other. And I thought this was like slam dunk. Like, this is like perfect genomic epi. Like, we're going to find like where it came from and like map it out. No. No. So all the strains were like wildly different from each other. Like, nothing really makes sense, except that we apparently just have like a very diverse pool of steno. So there was no like smoking gun. Even though the infections happened like right next to each other within a week, it's not the same strain. So equally powerful at being able to say, two cases are related is being able to say, no, it's not the same strain here. So we sort of have more of a, I guess, like systemic problem in terms of like creating a environment where steno can really grow. I thought I'd kind of nerd out on some like Carsi related stuff. So my group does like the embarrassingly parallel type of computing, where it's like, we have a workflow and we need to do it like 1,000 times. And so, I mean, I'm not even that old, but Nextflow has been like an absolutely amazing game changer for being able to like containerize and deploy these things at scale. And so, I don't know, I think Nextflow is more of like a biology-ish type environment, but you essentially can kick off workflows. It can access all sorts of containers. It can then sort of interface with a large variety of sort of batch submission script, processes. They've got pipelines for like, almost anything now. So this is their NF core, which is sort of like a repository. There's 128 in here. We do a lot of metagenomics in my group. There's a fantastic pipeline for that. This is what we were running all the time for SARS-CoV-2. So it sort of like breaks down all the pathways. I'd like to give a shout out to this pipeline, Bactopia, which is what we run basically on everything. So this was, basically, if I had to create a pipeline from scratch, this is what it would look like. And it's run by my friend who works for the Wyoming Public Health Lab, but you essentially can kick off, I think I processed 2 weeks ago, 1,200 bacterial genomes with this pipeline, like downloading the reads and processing them from the raw data. And it took like 20 hours, which is unheard of. And then we were just ready to rock. So thank you to my group and my students and postdocs and techs and everybody else, Carsi, of course. Thank you all. Any questions?
I had a quick question.
Yeah.
In the Nextflow pipeline, are you submitting them as a user in that diagram, or are you using a GitHub push for?
Yeah, so there's usually like a sort of like master control script that we SBatch, and then it will then auto submit to the queues.
Cool.
Yeah.
Any other questions? Yeah.
Yes, sir.
What's the best hypothesis about where the center for most came from? For that pool of them, it's just an environmental thing. Because of the water.
Yeah, it's a honestly, Vince, I don't know. like it's one of these bugs that like resists cleaning products as well. Like, we found it on ventilators. We found it on like the heater wires that were on ventilators. And it's just like one of these bugs that like is super good at resisting both antibiotics and cleaning. I mean, thankfully, it's not like I mentioned, it's not super pathogenic, so like yet, but meaning that you probably have to be pretty immunocompromised for it to actually cause severe infections. But like if you're being intubated and you have a tube, going right to where steno can grow, and like which was what was happening. I mean, it's like, okay, game on. And so but yeah, it's something that has perplexed us, because we keep finding it. NIH didn't seem interested in helping us find more of it. But
Thank you very much.
Cool. Thanks, everybody.
Heather?
Sure.
Yeah, I'm gonna get your presentation going.
Okay.
Heather's getting her water bottle. Daryl, is that trade-off between virulence and resistance, is that a fundamental property where there's some, it has to give up some mechanism? Yeah.
You know, like, that has been the historical thinking. Like, there's some fitness cost to resistance. Yeah, and I think that's kind of starting to be overturned. Like, you know, especially that a lot of these variants or resistant genes are on plasmids, and like the thought is like carrying the burden of these big plasmids through generations would eventually, I think, kick them out.
Yeah.
But that seems to not be the case.
Okay.
Definitely not.
Thank you.